I will start talking about chromatin. We heard yesterday about this topic and learned how much important is the presence in the chromatin of chromatin remodeling machines and in general factors that allow the nucleosome to move along the chromatin. What I'm going to talk today is about HMG proteins, which is a group of proteins involved in chromatin organization and that in general contribute to several processes which occurs within the nucleus by opening the chromatin fiber. In general, keep more open the chromatin fiber. We are more interested in HMGA proteins. Three factors belong to this family, the HMGA1A and the HMGA1B, which are the product of the splicing of the same gene, and HMGA2 that, although related to HMGA1, is the product of a different gene. The peculiar feature of this factor is the presence of the AT hook. This domain allows these proteins to interact with DNA, in particular with AT-rich stretches in the DNA, and they interact in the minor groove. By doing so, HMGA proteins can bend DNA and can also recruit factors on these sequences. And this, in general, allows the recruiting of other factors that can remodel the chromatin and can contribute to transcription. In general, the contribution HMGA proteins give to transcription has to be seen at two different levels. One, which is more specific at the gene-specific level, since they are able to, as I told, bind DNA, facilitate the binding of transcription factors to DNA, and by doing so, they form complexes which are called enantiosome and promoting transcription. Then they are able also to interact with the transcription factor independently on the binding to the DNA, and by doing so, they can modulate the affinity of this factor to DNA sequences, promoter, enhancer, and so on. The other level of regulation on transcription, these factors have on the chromatin, in the sense that they can bind to the DNA competing with the histone H1, and the result is a more open chromatin fiber. For this characteristic, these factors are considered and are called architectural transcription factors. It is interesting that HMGA are expressed at high levels during embryogenesis, and their expression decreases during differentiation, reaching very low levels in terminally differentiated cells. Very, very low levels, or sometimes are always almost undetectable. But following neoplastic transformation, the expression is again very high. The contribution these factors give to the embryonic development is underlined by the knockout of these genes in mice, which produce consistent phenotypic alterations and impair development. They also regulate the expression of specific genes in differentiated cells, and they also contribute to senescence. In fact, they contribute to the establishment and maintenance of senescence-associated heterochromatin foci. On the other hand, they contribute also to some pathological functions. They are overexpressed in tumor cells at high levels, and this is an old observation, more than 20 years ago. But the overexpression of these proteins in tumor cells is become a hallmark of cancer. In fact, almost all tumors express high levels of these proteins. Therefore, they are considered molecular markers of neoplastic transformation. In addition, they contribute to this process because transgenic mice overexpressing these proteins develop tumors. So apparently, HMGA perform different functions within the cells. They can have either pro-oncogenic function, anti-oncogenic function. That depends essentially on the molecular context where they are expressed. So it is crucial for understanding the function of these proteins to identify the molecular partners. So in general, to identify molecular partners, you can follow different approaches. One is the so-called hypothesis-driven approach, where interaction studies are prompted by, for example, the co-presence in close proximity of HMGA and specific transcription factor at the level of DNA regulatory sequences such as promoter and enhancers, or by overlapping biological function. And another approach, which I would call holistic approach, unbiased approach, relies on, for example, the Yeast-2 hybrid or protein array or proteomic approaches such as two-dimension gel electrophoresis coupled to blot overlay or affinity chromatography. Both of these approaches need later identification of the partners by mass spectrometry. We started a few years ago to identify molecular partners of HMGA using the hypothesis-driven approach. And what we have done was just to test in GST pull-down whether HMGA were able to bind nuclear proteins that were associated with tumor progression and tumor transformation. And we identified different proteins. For example, one, it is reported here, is P53. What we demonstrated was that HMGA1 was able to bind the entire family of P53 members, and this impaired the function of P53 in thyroid tumor cells. Okay, so what comes out from these approaches? Let's say that from our approaches and also from other labs, what it comes out is that there are several transcription factors that can interact with HMGA1, and therefore HMGA proteins have a role in transcription. Okay, but what happens if we look at an unbiased way at the interactors of HMGA? So what happens if we use, for example, other approaches to identify molecular partners? We used several approaches. What I want to show you now is just the last one that we used, which is a blot overlay approach. This is essentially schematized in this slide. So first, nuclear proteins are separated in the first dimension gel according to their isoelectric point, and later they are separated on second dimension for their molecular weight. Then proteins are transferred to a membrane, are incubated with HMGA1 protein, and then the complex is detected using a specific antibody. So at the end, you have a signal that allows you to identify molecular partners. We have followed this approach, and as you can see, we identify several potential interactors, but since the pattern was quite complex, we decided to prefractionate the nuclear extract using HPLC, and we separated in 4 different fractions, and we have done blot overlay for each of them. Here you can see, for example, the first fraction and the second fraction. So the idea is essentially that from the autoradiography, you can go back to the red ponceau stained membrane, and you can identify the spots on a gel that has been run in parallel and has been stained with Comassi or silver staining. Then the protein is identified through mass spectrometry. Using this approach, we have identified, for example, 60 spots in the F1 fraction, and we have done the same for the others. Later, we validated the interactions using a high-resolution 2D gels, which is called also zoom gel. And as you can see in this example, we could validate all the partners identified. To validate the approach, we selected some of these partners, and we have done GST pull-down experiments. And as you can see here, all of the protein tested interacted in GST pull-down with HMGA proteins. We also validated this interaction in vivo using co-affinity purification and mapped the interaction domain in HMGA protein. Okay, so what's the result of this approach? The result of this approach is that in addition of transcription factors, HMGA can bind, can associate also with other nuclear proteins. Proteins which are involved, for example, in DNA repair, proteins which are involved in chromatin structure, in mRNA processing, enzymes which can modify HMGA proteins, and other which are not classified. So what are we doing now is try to investigate on those which are more interesting to see whether we will be able to add new function to those which are already known. I have to say that up to now, the factors that we are working on have been confirmed in vivo, and also they give, the interaction have functional significance. 1 minute left. Okay. Since time is short, the next question was, okay, so HMGA establish a network of interactions in cancer cells. So is this network of interaction critical for cancer cells? So in other words, what happens if we perturb this network? For example, by silencing the expression of HMGA1. So we developed inducible cell lines which are inducible for silencing of HMGA1. And as you can see, we were able to efficiently silence the expression of HMGA1. These are the control. And then we look at the morphology of these cells. And as you can see, HMGA1 depleted cells grow more attached one to the others. They grow more regularly, and also they are larger and more flattened compared to the control ones that are growing one over the others. They are more prolonged. They have more fibroblast or mesenchymal aspect. This can be better appreciated if we look at the F-actin organization. In particular, at a higher magnitude, you can see that the distribution is essentially periplasmatic, and there are not stress fibers which are instead present in the control cells. What is more interesting is the 3D morphology, because cells, through the progression, the neoplastic progression, lose the polarity. And, but this cannot be appreciated in 2D cultures, and therefore we grow cells in matrigel where they can form colonies. And you can see the difference is quite dramatic. In fact, HMGA1 depleted cells can form these spheroids, and which are very regular. Instead, control cells do not. They proliferate, they form these colonies which are very irregular, and they have this stellate projection. We measured the growth rate, which is about the same until they reach confluence, but then HMGA1 depleted cells grow slower, and this suggests then the contact inhibition is at least partially restored. Then we evaluated the Anchorage-independent growth by growing cells in soft agar, and as you can see, the difference is dramatic. And also with the wood healing assay, we test motility, and again, as you can see, the depletion of HMGA1 protein in these cells cause a decrease in motility. So HMGA1 in these cells also impairs motility. And then, okay, I'm going to conclude then, just showing that also migration invasion is impaired. And more importantly, in vivo analysis tells that even though there are preliminary data, the mice that were injected in the mammary fat pad developed tumors which are much smaller than the control ones, and what is more important is that they do not develop or develop few metastases, while the control develop, almost all of them develop metastases. Microarray experiment, let's say the result of the microarray experiment is consistent with the morphological changes that we observed. So in conclusion, I would say that altogether these results allow to demonstrate that HMGA establishes a network of interaction in tumor cells, which is critical for promoting invasion and metastasis. And in the last one, I would really like to thank all the people that has done the work in the lab, Riccardo, Silvia, Laura, Ilenia, Gloria, and Elisa, and also the collaboration that we have with the other labs. Thank you very much.